This video is Unit 2, the second video on photosynthesis. And this one will primarily be focusing on the light-dependent reaction of photosynthesis. Just a short review on electrons. Remember, there are things called valence electrons. These are the electrons found in the outermost shell of the atom. These are the electrons that are most likely to go if there's some sort of interaction between other atoms. The atom is most likely to let go of those, particularly in like an ionic bonding situation, or if there's a lot of energy. And so if you take magnesium, for instance, it has 2 electrons in its valence shell. That's going to be important as we look at this reaction. As energy is applied to that magnesium, and energy is more and more energy is given to that magnesium, those electrons are going to get further and further away from the nucleus of the atom, causing it to eventually let go of those electrons more readily. And so here's the light reaction for photosynthesis. Again, water is a reactant on this end of things. Oxygen is going to be the byproduct here. But again, the most important products of this part of the reaction are ATP and NADPH. ATP, adenosine triphosphate, is the energy currency of the cell, and NADPH is considered an electron carrier. Those electrons represent energy. It's going to carry those electrons to a place where it can get rid of them and use them. There are 2 different electron flows in the light-dependent reaction. The one that we're going to spend most of our time on is the non-cyclic flow, meaning that those electrons have a starting point. That starting point is going to be photosystem 2, as you see on this picture, and the end point is going to be NADPH. They're not cycling through. It has an end point that's not the beginning. There's also a cyclic electron flow, and we aren't going to spend any time on this other than to say, in the cyclic electron flow, the purpose of the cyclic electron flow is to make more ATP. That's all you need to know. Don't worry about its mechanisms. Don't worry about anything else. There is one important thing here. Cytochrome C, or cytochrome complex, there is a protein that is found in most organisms, and it is one of those things that you can look at and find a common ancestor or ancestry with organisms. But other than that, there's nothing important here. We're going to spend most of our time looking at the non-cyclic flow of electrons. And again, this is occurring on the thylakoid membrane. So as we talk about this, remember that we're dealing with a thylakoid. I'm going to draw a picture of a cell membrane, and that membrane is not a cell membrane. It's not the outer membrane of the cell. It is the thylakoid membrane. Now, if you're a bacteria and you're doing photosynthesis, that will be on your cell membrane. But for our purposes, we're going to pretend we're in a plant, and that's what we're doing. And just a note too, the chloroplast, lots of thylakoids, lots of surface area. This is happening all over the chloroplast. There are lots of chloroplasts in a plant cell. And so this is happening a lot. And so I'm going to just be showing you one snapshot of this, but know that this is happening all over the plant cell. And so with that, I want to draw a picture of the thylakoid membrane. Remember, on the thylakoid membrane, there are these photosystems. I'll just draw these as these big blocks because it's easier to do it that way. Sizes, proportions, none of those things are that important here. Here we have this funky looking protein. This is ATP synthase, and this is photosystem 2 and photosystem 1. And then one more thing to label here, the portion between photosystem 2 and 1 is called the electron transport chain. A couple other things to note. Out here on the top portion of my screen is the stroma. Inside here on the bottom portion, which is the inside of the thylakoid, is called the lumen of the thylakoid. And just note, you go to the website or you look in a textbook, this reaction is going to look a lot more complicated. There are a lot more steps. A lot of these little proteins have names, and we're not going to learn any of that because that's beyond the scope of this class. And so what I mainly want you to focus on is what these electrons are doing and how they're used to make both ATP and they're captured in this electron carrier called NADPH. So in order to symbolize the presence of these electrons, I'm going to draw this green blob on the top of these photosystems because inside the photosystems are chlorophyll molecules, and those chlorophyll molecules have magnesium in their center, and in that magnesium are two valence electrons, which are the key to all living things. And to symbolize light, I'm going to draw some lightning because lightning's cool. Don't get confused though, it's actual sunlight. Okay, so light hits photosystem 2. When that occurs, photosystem 2 is going to, the chlorophyll there in photosystem 2 is going to let go of those two electrons. That magnesium is going to get excited. It's going to let go of those two electrons that it has, and it is going to release them. And those electrons are going to move into the electron transport chain, or the ETC. It does exactly what you think it's going to do. It transports electrons. Now, one thing that's important in the transport of these electrons is there are these reactions called redox reactions. Redox reaction is a lot more complicated than we're going to talk about it in here, but it's basically two molecules exchanging electrons. And so on this electron transport chain, you're going to have a series of proteins that we haven't drawn, and they're going to be exchanging electrons. As a molecule receives an electron, it becomes reduced. Think about it. You're receiving two negatives, your charge is going to be reduced. As it gives those electrons away, that losing of the electrons is called oxidation. And so we reduce that reduction oxidation by just calling it redox. And what these redox do, these excited electrons moving down this electron transport chain, you can think of like a game of hot potato. They're passing off these hot excited electrons, and as they do it, the electron transport chain is going to be energized. And that energy is going to be able to be used to do some cellular work, which we're going to look at. All right, so we'll talk about that work in just a moment. Now, magnesium's lost its two electrons. It doesn't have any more valence electrons. And so in order for this reaction to continue to happen, we're going to need a steady stream of those electrons. And that is where water is going to come in. This is why plants need water. Water's going to be there on photosystem 2. The light is going to interact with water, and it's going to basically shatter the water molecule. Now, water is made up of several parts. Water is made up of an O, and it's made up of two H's. And what are H's made up of? Two hydrogen ions and two electrons. And so essentially 5 parts to that simple little water molecule. You know, hydrogen loves to get rid of its electron more than anything on Earth. And so those electrons are going to be easily spared from hydrogen. Those hydrogen ions, or also we'll just call those protons, are going to be released as well. And so I'm going to show you that here. Those two electrons, I'm just going to draw that here. They're going to be continued to be fed into this chain, almost like the steady stream of electrons. Oxygen is going to be given off as a byproduct at this point, which satisfies one of the products of this reaction, right? And then those protons, they're going to begin to build up out here in the stroma. Right. So I'm going to build these protons up. I'm going to leave them here for a moment. Those electrons, actually, we're talking about them right now. The electrons passing through the electron transport chain, that series of redox reactions passing electrons off, that is going to power the movement of these protons into the lumen. And what is that going to do? That's going to build up a concentration gradient inside the lumen, whereas we have a high concentration of protons in the lumen, a low concentration of protons outside the lumen, and so that concentration gradient is going to be able to be used to do work. So the energy of the redox reactions is used to actively transport protons into the thylakoid. Once those electrons do that, once they get to photosystem 1, they're a little tired from their journey. And so they get re-energized here at photosystem 1, and they're going to get passed off to our friend NAD plus, which is converted to NADPH, and it's going off to the Calvin cycle. So here, NADPH, one of the products that was supposed to be made in this light-dependent reaction. Now, what can we do with these protons here that have built up in the lumen? A lot of cellular work uses concentration gradients. Well, this one's no different. In this particular version, these protons are going to diffuse back across with being facilitated by this molecule ATP synthase. What that causes ATP synthase to do is literally spin around. Think of like a top or something spinning around. Transfers the energy of that diffusion into mechanical energy, causing ATP synthase to spin around. That spinning around, I think I have a picture of this here, yep, is going to create some kinetic energy in order to make ATP. Now, ATP synthase is one of the most complicated things in the cell. It's extremely complex. It's this little motor, basically, that works, and very complex protein. Strongly encourage you to look at that on your own. But just believe me that these protons diffusing through it cause this molecule to spin around, and through that allows for the creation of, or the phosphorylation of ADP into ATP, and these ATP molecules that are created are headed off to the Calvin cycle. And so here we have both of these molecules, NADPH and ATP, that have been created in order to facilitate the creation of sugar in the Calvin cycle.